What is up, freshman biology?
This is Advanced Biotech bringing you a PCR tutorial.
Polymerase chain reaction, or PCR, amplifies a target strand of DNA exponentially, so scientists have larger samples of DNA to test.
Today's lab is trying to find out who committed a crime using a sample of their DNA that was found at the crime scene.
So before you step foot in the lab,
here are some things that you shouldn't do.
Put a spell on you. Put a spell on you. Put a spell on you.
Lab procedure.
Yeah, definitely not. In fact, you know how much stuff that was all over here.
Oh, quite.
It would be pretty cool, but
I feel like it would be cool.
little tiny incisions that they just take like practice.
Alright, so now let's actually get into what we're supposed to be doing. First, we labeled each microcentrifuge tube according to which DNA sample it would hold.
each lab table was assigned a different color microcentrifuge tube that corresponds to the sample of DNA.
Each sample of DNA was picked up either from the crime scene or taken from the suspects. We micropipetted 10 microliters of the DNA samples into the corresponding microcentrifuge tubes.
Put a spell on you.
Our group was assigned the DNA picked up at the crime scene,
Put a spell on you.
as well as the DNA taken from suspect D.
Put a spell on you.
Alright, so this is a micropipette. The numbers in the window correspond to the volume it will pick up.
Yes.
Make sure to close the lid on all tubes just in case you spill something.
This is the process to make agarose gel plates.
To begin making an agarose gel, you begin by pouring 3 grams of agarose powder into a beaker.
The thrill is gone. The thrill is gone. The thrill is gone.
After adding your agarose powder, add 100 milliliters of TAE buffer into the beaker and stir until mixed. Remember to always add liquids to solids when you're mixing in a lab.
After mixing, take the solution to the microwave. Here you will heat it in increments of 15 seconds, stirring between each. Continue to heat and stir in increments until the solution is clear with no streaks.
Look around you. Change your heart.
While they were heating the agarose solution, I went back to our lab table to set up the gel plate molds.
Change your heart. Look around you. Change your heart. Look around you.
Okay, so here I throw all lab procedure out the window and blatantly disregard the hot glove that Lexi has literally put right in front of my face.
If distributed evenly, each solution should fill 2 gel molds.
As you can see, we very obviously poured ours light just to be safe because we were sharing with another group.
But Jacob decided he needed to use the rest of the solution, leaving us with a very thin plate.
Well, Jacob used the rest of it, so.
Sorry.
Look around you.
While the plates were setting, we began to label our PCR tubes in preparation for filling them with the corresponding DNA sample.
Look around you. Like the sunshine, change your heart. I need your loving. Look around you.
This is us just labeling them really fast. So, I mean, you can enjoy if you want, but like I'm not gonna make you enjoy this. So, alright.
Here we fill the PCR tubes with their respective DNA sample. In our lab, we have 4 different sizes of micropipettes.
This is the 0.5 to 10 microliter pipette. The red band matches with the clear tips. Next is the 2 to 20 microliter pipette. The yellow band matches with the yellow tips. This is the 20 to 200 microliter pipette. Again, the yellow band corresponds with the yellow tip. Finally, our biggest pipette is the 100 to 1000 microliter pipette. The blue band matches with the blue tips.
The dial on the pipette allows you to change the volume of the liquid it will pick up within the range of the numbers shown on the pipette.
At the top of the pipette, there is a button with 2 stops. The first stop pushes the liquid out of the tip, while the second stop forces any leftover residue of liquid out of the tip.
On the side of the pipette, there's another button used to discard the used tip. Always change tips when changing liquids as to not contaminate the samples.
Keep in mind that you're never supposed to place a micropipette on its side when there is liquid in the tip. It will completely ruin the inside of the pipette, making it unable to function.
Why?
So you were
Sounds like we were fine.
So the jet engine-like sound you hear in the background of this video is a microcentrifuge.
A microcentrifuge mixes samples by spinning at an extremely high speed, also known as centrifugal force. Always balance the microcentrifuge by placing tubes directly across from each other, or it won't mix samples correctly.
Always let the microcentrifuge completely stop before you open it up, or else things will fly everywhere and it's just not good, I promise. So again, this is us just doing something really fast, and that's the fire alarm, so I guess we'll be right back. Okay, so after that momentary interruption, this is just us pipetting really fast.
You'll know the agarose has solidified when it turns cloudy. Then label a plastic bag with the group members' names. Gently place your agarose in the bag as to not rip it, and place it in the fridge.
In our lab, we label everything. In this case, it's because people are dirty thieves and they will steal your agarose gel.
Before we started our lab, our group programmed the thermocycler. The thermocycler is a machine that helps amplify DNA by cycling through various temperatures.
We then placed the DNA-filled PCR tubes in the thermocycler. So after the thermocycler wouldn't turn on, I realized that I had actually plugged in the microwave, so I unplugged that and actually plugged in the thermocycler. We set it to the right program and we began the cycle. This is us setting the cycle. Again, I know, super exciting.
Complete.
Alright, welcome back to day 2 of our PCR tutorial. Today it goes down when we run the samples in the electrophoresis chamber and find out who committed the crime.
So, what happened? What crime?
You know, I really don't know. We're just out here running the samples. Honestly, nobody tells us anything.
After receiving our samples back from the thermocycler, the next step is to micropipette 20 microliters of loading dye into the PCR tubes.
This time I'm asking you to please enjoy this part because it was as much fun as it looks. Alright, so here you can see how the loading dye changes the color of the master mix DNA sample collection thing. Pretty dope, sick, nasty, I know. Like, you don't have to tell me I did this myself. Those are my hands. I know it looks cool.
This is an electrophoresis chamber. It emits an electric current that causes the negatively charged DNA to run towards the positive side of the chamber.
We then placed the agarose gel in the chamber with the wells on the negative side. We poured the TAE buffer in the chamber until it covered the gel and wet loaded 20 microliters of the DNA samples into the gel.
Wet loading is exactly that, micropipetting DNA samples into the gel mold after the buffer has been added. Make sure to write down which DNA sample is in which lane.
In lane 1, we placed an allele ladder. An allele ladder is made up of different sized base pairs that are evenly spread out that we use to measure how far the DNA has traveled through the gel. In lane 2, we placed the crime scene DNA. Lane 3 was suspect A's DNA. In lane 4, we placed suspect B's DNA. In lane 5, we placed suspect C's DNA. And in lane 6, we placed suspect D's DNA.
Make sure the wells are over the black line that indicates the negative side of the chamber. Plug in the chamber to the power source before turning it on to avoid certain death. Next, set the power source to 100 volts as well as a timer for 30 minutes and turn on the power source. If baby little bubbles appear at both ends of the chamber, you know it's working.
Now sit back for what feels like the longest 30 minutes of your life. Like, it's agonizing. It seriously feels like death would come faster than the chamber runs the DNA samples. So after the chamber was done running, right here is where you see me very obviously struggle super hard to get the lid off. I don't know, I couldn't do it. I just don't know, I don't know.
Once the 30 minutes is up, turn off the power source and unplug it from the chamber. Gently remove the gel from the chamber and set it in fast blast dye for 5 minutes.
Now sit back for another agonizing 5 minutes while the gel stains. Next is a slight armor cap. You place the now blue gel in a bucket of warm water and very gently rock back and forth for 5 minutes. Once the 5 minutes is up, you repeat the rocking once more.
Until you learn the meaning of the until you love the love you had to.
Now take the gel out and place on a paper towel for drying and observation. You should now be able to see bands indicating the base pairs of the DNA. Compare those to the DNA found at the crime scene. In our case, our criminal was suspect C.
Alright, and now we thoroughly hope that you have enjoyed this PCR tutorial. We had so much fun making it, and we hope you had just as much fun watching it. Thank you, and we'll see you back here again for another tutorial. Alright, and always clean up the lab, because you know a clean lab is a happy lab, you know what I'm saying?